Hello, in this video we will discuss about the insulin signaling pathway. So first of all we should need to understand about the insulin and basic background about the what is insulin. So insulin is the peptide hormone released from the pancreas to stimulate the anabolism. So the metabolism, the type of metabolism is anabolism. Anabolism mean the synthesis of glycogen, synthesis of lipids, and synthesis of the protein. So the absorption of glucose into the muscle and liver is going to the glycogenesis and lipogenesis. So in this way, here is let's begin to understand first of all the control of secretion of the insulin, and we will also understand about the mechanism of the insulin secretion from the beta cell of the pancreas. So let's begin to understand first. We'll begin to the fat state. So when during the eating time, so we will eat the some food contain carbohydrates and protein, which that go into the stomach in the form of bolus and digest the protein and go into the duodenum, and further digestion of the lipids and amylase as well as the lipids, amylose and lipids. Oh, sorry, not amylase, because amylase enzyme used for it. So here is the enterocytes are present in the duodenum, and here is the brain, and this is the pancreas. Pancreas contain islet of Langerhans. I am drawing here is just three cell for understanding type of cell: alpha cell, beta cell, and delta cell. Delta cell is responsible for the production of somatostatin, while the alpha glucagon, beta insulin. So here is the GLP-1 glucagon like peptide 1 will trigger amino acid and glucose and fat are present if in the duodenum feels stretching to release from the enteroendocrine cell to stimulate and inhibit the alpha cell for the production of glucagon. So here is the vagal nerves which that stimulate, and that is basically used for the parasympathetic nervous system of the autonomic nervous system, and sympathetic nervous system will be inhibit while the parasympathetic nervous system will be stimulated to trigger this nerve, and this nerve will bind and to release the somatostatin by the neurotransmitter through synapse. So the cellular synapse with the delta cell, and in this way here is the neurotransmitter we release to bind with the neurotransmitter receptor are present in the delta cell to produce somatostatin. This somatostatin will bind with the alpha cell. After the binding with the alpha cell to trigger the releasing of the alpha cell, trigger to the paracrine hormone, which that will be triggered to the beta cell to release the insulin. After the releasing of the insulin is releasing of the insulin, insulin will get target with a different tissue, for example liver and muscle. Muscle and liver contain an insulin receptor. When insulin will bind with the liver and muscle, and the GLUT transporter channel 4 will be open. After the opening of the GLUT transporter channel 4, so the glucose will move inside and to become a synthesis of the glycogen from the glucose. And in the case of the adipose tissue, the lipogenesis will be occur through glucose. So here is that during eating time, during fat state, the stimulate to pancreas for production of insulin. So important thing is that when we will take the glucose and protein will absorb into the stomach, and after this basically go into the bloodstream. After the bloodstream, hyperglycemia will be occur, and here is the pancreas which that produce the insulin into the blood circulatory system. After the production of the insulin into the blood circulatory system, so the insulin will bind with the insulin receptor with a different tissue where the desire of the glucose, so the glucose requirement will target the tissue, insulin target to the tissue where the requirement of the glucose is more. So let's begin to understand. Here is the adipose tissue. This adipose tissue contains a channels. This channel GLUT4 transporter channel will open through a insulin binding with the insulin receptor to enter the glucose to lipogenesis. So here let's begin to understand the mechanism of the releasing of insulin from the beta cell. So here is the diagram showing that is the channel. So there are different channel and nucleus are present, and this is the insulin vesicle. After the synthesis of insulin from the transcription to translation from the nucleus. So I am not describing about the synthesis, but I am just understanding for the releasing mechanism. So here is, if you want to watch the whole video, so I have video you can search. And here is the glucose, and this is the glycolysis will occur, and in this way production of ATP will be more. So the ATP dependent calcium channel will be closed due to the enhancement of the ATP. So the GLUT2 channel is requirement for the entering of the glucose, and after this the depolarization will trigger the calcium channel to influx of the calcium channel will trigger the efflux or exocytosis of the insulin will bind with the cell required cell. So after this binding, the glucose will move inside. So let's begin to understand the whole process in the molecular biology. So we will discuss about the signaling pathway. Let's begin to understand. So here I am drawing the plasma membrane for understanding with the receptor, insulin receptor. This insulin receptor will bind with the two molecules of insulin to conformation chain. After the changing of the conformation, so the insulin receptor substrate will be bind with the insulin receptor into the intracellular domain, while the extracellular domain contain a insulin molecule. So after this insulin will be bind, so here is the insulin receptor substrate will be bind, and after the insulin receptor substrate bind to recruit more protein for further triggering of the molecular pattern and molecular signaling. So here is the SH2 protein also bind with the growth binding protein 2 bind, and source will be bind after the recruitment by the phosphorylation to stimulate and trigger the RAS inactive to become a RAS activate by the GTP, and this GTP will bind and recruit the RAF protein. After this complex formation is known as the dimerization. So this dimerization will trigger the in the presence of MAP kinase MAP activated mitogen activated protein kinase will trigger antiphosphorylation of the mitogen activated uh, protein kinase 1, and it's in this way here is the activation by phosphorylation to the MAP kinase will trigger the ERK and MAP kinase pathway. This ERK and MAP kinase pathway will trigger the transcriptional factor phosphorylation of the transcriptional factor. After this transcriptional factor, these transcriptional factor will go into the nucleus. After the movement into the nucleus through a nuclear pore, and here is the nuclear pore is channel like structure, and here is the inside of the nucleus is here is the chromosome, and this chromosome contain DNA. I am describing with the DNA and bind with this transcription factor with the DNA, and after this DNA is basically is the uh, uh, PGSK3 gene, which that is known as the glycogen synthase 3 kinase. This gene will produce the glycogen synthase 3 kinase, and here is the another protein is known as the Castias B lineage lymphoma protein C, also transcribed, and the RAS and MICE gene is also activated for the further proliferation as well as the cell division and cell growth. So remember, here is another mechanism. In this way, the recruitment through a insulin substrate receptor for recruitment after the phosphorylation with the P85 protein and protein 110, and it is also bound with the phosphoinositide 3 kinase, and this phosphoinositide 3 kinase with PIP2 phosphoinositide 4,5 diphosphate bisphosphate, And here you can see the structure I am drawing, which that will be phosphoinositide 3 kinase is used to produce the PIP3 phosphoinositide triphosphate. So this triphosphate will be formed. After that, three phosphate group attachment by phosphorylation and the recruitment of the AKT protein. After this AKT protein will be bind and to dissociate to form a PAKT. So the phosphorylazed AKT, and this will be inactive PGSK3. PGSK3 inactivate to activate. So the PGSK3 activation through a transcriptional factor as well as the inactive enzyme will become active. So on the other hand, here is the mTOR pathway. mTOR pathway is also triggered through a AKT protein. And on the other hand, the fork head transcriptional factor. This fork head transcriptional factor will inhibit of gluconeogenesis, and the pattern as well as the set of the gluconeogenesis related enzyme will be inhibited through a transcription inhibition, while here the PGSK3 will be inactive to become an active PGSK3 glycogen synthase 3 kinase and to become a glycogen synthase, and this glycogen synthesis very important for this mechanism. While mTOR will be a more proliferate and more trigger the ERK1 protein, which that is a transcription factor, and while the glycogen and cell division and for protein synthesis is very important and produced by this mTOR triggering pathway. And here the active E1F2 is also trigger the protein synthesis through a glycogen synthase 3 kinase is also triggered. While here is the CBL, which that is bind the CBL associated protein with the CRK, and on the other hand the C3G recruitment after binding by the phosphorylation, and TC10 will trigger the GLUT4 vesicle. After the triggering of the GLUT4 vesicle will bind with the plasma membrane for the influx of the glucose. So the glucose will move inside for the glycogenesis, so the glycogen formation. But in the case of another pathway is very important. After the PIP3 will be dissociate into the IP3 inositide triphosphate, and after this IP3 will trigger the endoplasmic reticulum to increase the calcium level to recruit the PKC protein kinase C will bind with the protein kinase C with DAG diacylglycerol to proliferate and the synthesis of protein. So thanks for watching. Please make sure to subscribe, like and share, give the feedback, and if you like this video, thumbs up. Bye.